Hello everyone. Still confused between MHC1 and MHC2? Why do some cells shows antigen to CD4 and others to CD8? Well, let's clear it. By the end of this lecture, you will master MHC like never before. Now, let's start with the definition first. MHC, that is major histocompatibility complex, is a group of genes. Now, what do these genes do? These genes code for proteins. These proteins are found on the surface of cells. Which cell? On almost every human cell. Almost all human cell. So, MHC is a group of genes which code for proteins, and these proteins are found on the surface of almost every human cell. That helps in the immune system in recognizing foreign substance. Now, let's understand how does it help. So, think of your MHC like a board, okay? MHC is a group of genes coding for proteins on the surface of the cell. So you imagine MHC like a board on the surface of the cell, okay? So what does this board do? It pick up tiny proteins or peptides, okay? Tiny proteins or peptides from inside the cell. If it's a healthy cell, okay, if it's a healthy cell, a normal cell, so it will take self protein, the self peptide, self antigen. Okay, this is the self protein, self peptide, and display it on the MHC board. Now, when your immune police, that is the T cell, will come for patrolling and will see this board, it will pass it as a healthy cell. Everything is fine. Now, let's see what happens when the same cell is infected by a pathogen, be it a virus, be it cancer, anything. So, what will happen? It will take the peptide of the fragment of this pathogen and it will display it on its board. And when your immune police, your T cell comes for patrolling, what will happen? It will look at this board and it will issue a intruder alert. Clear? This is what happens. So, these proteins, the proteins which are coded by the MHC genes, they do what? They bind the peptide fragments which is derived from either the pathogen or self and presents them to T cell, our immune police, for recognition, thereby playing a crucial role in immune response and transplantation compatibility. Now, in humans, these proteins are called HLA, human leukocyte antigens. You know why? Because it was discovered in human leukocyte. That's why it's called human leukocyte antigens. Okay. Now, let's move ahead. Now, just like every Indian citizen holds an Aadhaar card as a proof of identity, as a proof of citizenship, every cell in your body also carries its own Aadhaar card. That is the MHC molecule, okay, as a proof that they belongs to you, that they are residing in you, they are a citizen of your body, okay. That's why just like your Aadhaar card, your MHC molecule is also unique, okay. That means no two individual will have the same MHC. They can't have the same Aadhaar card, they won't have the same MHC, but we have one exception here. Two identical twins can have the same MHC molecule. Clear? Now, that's why during the transplantation, there is a mismatch because of this MHC molecule. Okay. Now, you see, bigger the mismatch, faster is the rejection. Now, tell you what does this mean? Think of it like we'll start with identical twins. Okay. One identical twin is the transplanting organ to is another twin. So, what happens here? They are sharing the same MHC. They have the same Aadhaar card. So, what happens here? There is very less chance of rejection, very low rate of rejection. Clear? Now, let's move to siblings now. Not identical twins, just siblings. Now, these siblings may share some same Aadhaar number. In your Aadhaar card, there is number. So, these siblings may share some Aadhaar number. So, the MHC will be a partial match. Okay. Now, with the help of medicine like immunosuppressants, you can still manage the rejection. It is still manageable. It will be there, but manageable. Now, we'll talk about unrelated donor. What happens when there is an unrelated donor? In such a case, the Aadhaar card, the MHC molecules are completely different. So, what will be there? There is a very fast rejection. Clear? You understood this statement? That means bigger the mismatch, faster the rejection. Clear? Now you see, what is this MHC doing? MHC is deciding the compatibility between the donor and the recipient. Are they compatible enough to donate organ, to share the, to transfer the organ? So that's why, because of this, it's also known as histocompatibility antigens. Clear? Understood? Now let's move ahead. Now before I go into the MHC genes and their product, let's quickly revise the 12th standard ka genetics. Okay. Now her somatic cell, I am telling the word somatic. Okay. Her somatic cell के nucleus में they have 23 pair of chromosome. Okay, हर somatic cell के nucleus में they have 23 pair of chromosome. In the male, the 23rd pair is XY and in the female is XX. Okay, now in the cell they have nucleus and they have chromosome. Chromosome is made up of DNA and DNA के एक segment को we call gene. Okay, gene is what? Gene is the segment of DNA. Now this DNA and this gene is made up of these base pairs. Okay, this is one base pair, this is the second base pair, like this. Okay, this is just to give you some context. जो भी आगे बात करेंगे उसके बारे. I hope it's clear. Now let's focus on chromosome 6. Why? Because on the short arm of chromosome 6, it has two arms, one long arm and one short arm. Now on the short arm of chromosome 6 lies the HLA complex, which has the gene, MHC gene, which makes the MHC protein, the MHC molecules. Understood? Now you see this part is the one which has this HLA complex, right? Now this HLA complex, this whole chromosome 6 is made up of almost 170 million base pair. Okay, 170 million base pair, this whole chromosome 6. And this part of HLA complex, it extends over 4 million base pair. Okay, so this whole chromosome 6 is made up of 170 million base pair and the HLA complex ये फैला हुआ है over 4 million base pair. Okay, and in this area it is covering more than 100 genes. Okay, now these genes are further grouped into three region, MHC region 1, 2 and 3, which we will see separately. Okay, so let's move ahead. Now this is the chromosome 6. This is the short arm of chromosome 6. Okay, this is the HLA gene complex region. It is divided into three parts: HLA class 1, HLA class 2, HLA class 3 region. Clear? Now let's see what all genes they have and what all proteins it's forming. HLA class 1 region. Now remember, I told you it has more than 100 genes, but here we are only reading about and learning about the one which are important. Okay, we don't need to learn about all the more than 100 genes. That's not possible. So we'll only be talking about the one that are important. Clear? So your HLA class 1 region has the gene HLA-A, HLA-B, HLA-C. They will form protein. Which protein they will form? They will form HLA protein A, HLA protein B and HLA protein C. Okay, now these proteins HLA-A, B and C are going to make the alpha chain of MHC1. Okay, HLA class 1 makes the alpha chain of MHC1. Now this alpha chain has three domains: alpha 1, alpha 2 and alpha 3. Okay, now this alpha chain alone is very shy. It's not able to express itself. So in such cases, what do we need to do? We will get help from a neighbor chromosome. So your chromosome 15, okay, your chromosome 15 has some genes. Every chromosome has genes, right? So chromosome 15 has genes which are non-MHC, because MHC genes are only present on this part of chromosome 6. So your chromosome has some non-HLA gene which forms beta-2 microglobulin. Okay, now this beta-2 microglobulin comes and attaches here and together they form MHC1. Now beta-2 globulin is helping this alpha chain to express itself. Clear? Now think of this as this structure. Okay, think of this as this half-eaten bread with a chocolate sauce. Okay, so this is the alpha 1, this is I have taken a bite, this is the alpha 2, This is the alpha 3, and alone this bread is very dry. If I've taken a bite, it's very dry. So if I dip it in this chocolate sauce, what will this chocolate sauce do? It will enhance. It will do what? It will enhance the flavor. ठीक है? ये फ्लेवर को और एक्सप्रेस कर देगा. So this is our beta 2 microglobulin. Okay, now see here. I have taken the bite here between alpha 1 and alpha 2. So here is the groove. Now what is this groove for? This groove is for antigen peptide to come and bind. What antigen? Antigen which is affecting the cell on which this MHC molecule is present. Understood? Clear? Now this MHC class 1 molecule is present on which cells? It is present on all nucleated cells. Okay, it is present on all nucleated cells and platelets. Now there is exception to this. It is not present in your RBC and it is not present in your sperm cells. Clear? This is your HLA class 1. Now we'll talk about your HLA class 2 region. Okay, so in 1 we read A, B and C. So in 2 we will move to D. Okay, so this is gene here are DP, DQ and DR, and the protein which is formed here are HLA-DP, HLA-DQ and HLA-DR. Clear? Now these proteins are forming what? These proteins are forming the alpha and beta chain of MHC2 molecule. Okay, this was for MHC1 and this is for MHC2. Clear? For your MHC2 protein. So your HLA class 2 region with the proteins of this HLA class 2 are forming both alpha and beta chain of the MHC2. Now you think of this as two friends holding hand, okay? A girl and a boy holding hands. This looks like this. This is a boy and this is a girl. The boy is the alpha, okay? It's the alpha 1 domain and this is the alpha 2 domain, and girl is the beta 1 domain and beta 2 domain, okay? They are holding hand which has a groove present. This is the part where the antigen will come and bind. The antigenic peptide will come and bind. This is the cleft, okay? Now you think of it like that. Whenever you have a confusion, always remember this picture of this half-eaten bread with chocolate sauce and these two friends holding hand. For your MHC1, if you just remember this half-eaten bread, alpha 1, alpha 2, alpha 3 and beta 2 microglobulin. Where was the antigen binding? In MHC1, it was binding between alpha 1 and alpha 2. Okay, there is this big bite which is here. Okay, now coming to MHC2, where is the antigen peptide binding? It is binding between two friends holding hand. One is alpha, one is beta. So alpha 1 and beta 1. In between the alpha 1 and beta 1, there is the antigenic peptide. The antigen, it will come and bind. Clear? Understood? Now, on what all cells we have MHC2 present? MHC2 protein, this MHC2 molecule are present on antigen presenting cells. Okay. What are the professional antigen presenting cells? They are your B cell and your monocytes, macrophages and macrophages, B cells and dendritic cell. Clear? This is MBD, must be doctor. Monocyte, macrophages, B cell, dendritic cell. You can get a question like this. Out of all the cells on which your MHC2 molecule is present, and you won't get APC, antigen presenting cell, as an option, you will get one of the names. So remember, monocyte, macrophages, B cell and dendritic cell. MBD must be doctor. Clear? Understood? Now, this was your HLA class 1 and HLA class 2 region. Now, there is something known as HLA class 3 region also, but these are not related to any antigen presentation at all. Why? Because we have MHC class 1 molecule present, MHC2 present, but we don't have, MHC2 present, but we don't have anything known as MHC3 present. MHC3 molecule, MHC3 protein doesn't exist. So this HLA class 3 region is doing what? It is coding for proteins like your complement proteins. Okay, your complement protein, your tumor necrosis factor, your tumor necrosis factor alpha and beta, and your heat shock protein. Understood? Like your accessory proteins. Clear? Complete? Now we'll move to the structure of MHC. Now, your MHC class 1, MHC1 molecule, it's like this, half-eaten bread with this chocolate sauce. Always remember this diagram, okay? So, how many domains it has? It has alpha and beta. Alpha has 3 domains, alpha 1, alpha 2, and alpha 3. And the bite here is the peptide binding cleft, which was alpha 1 and alpha 2. Clear? Now, it is present on all nucleated cells and platelets except RBC and sperm cells, right? So this is the cell, a nucleated cell. On the surface, it is present. Now, it presents this antigen. Now, MHC1 presents only endogenous antigen. Remember this, endogenous antigen. Okay? To whom? To your T cells. Now, which T cell? It presents your antigen, endogenous antigen to your snipers, to your killer, to your CD8 cytotoxic T cell. Okay, CD8 killer cells, cytotoxic T cell. Now, see, which part of this MHC molecule will bind to this CD8 portion of cytotoxic T cell? See, this MHC1 molecule has this alpha 1, alpha 2, alpha 3 and beta 2 microglobulin is just for moral support. It is just helping the shy alpha to express itself. Apart from that, it has no function. Okay. Now, alpha 1 and alpha 2 are already busy with this peptide binding cleft with the antigen which is binding here. Now, what is left with us? We are left with alpha 3. So, alpha 3 will bind to CD8 of cytotoxic T cell, the CD8 molecule of cytotoxic T cell. Clear? Now we'll talk about MHC2 molecule. MHC2 molecule are like two friends, a girl and a boy holding hand. Clear? Guy is the alpha, alpha 1, alpha 2, girl was the beta, beta 1, beta 2. It was present on the surface of, this is the antigen presenting cell. Example of your antigen presenting cell must be doctor. Okay, monocyte, macrophages, B cell and dendritic cell. Okay, now you see here the antigen is binding in between when they are where they are holding hands, that is alpha 1 and beta 1. Okay, now your MHC2 molecule which is present on the antigen presenting cell, they present the antigen to your CD4 T helper cell. Okay, CD4 T helper cell. Now you see this CD4 T helper cell, the CD4 molecule of this T helper cell will bind to which portion of this MHC2? Now you think, understand here. Alpha 1 and beta 1 are busy with this peptide binding cleft with the antigen here. Now we are left with alpha 2 and beta 2. This alpha 2 is the alpha male here. He is like, I don't want to do any work. So what we are left with? We are left with our poor beta 2. This poor girl will bind to the beta 2 will bind to the CD4 molecule of the T helper cell. Clear? This was the MHC structure. Now, you know one more point here. In your MHC1 molecule, you see this is the alpha 1, alpha 2, alpha 3 and beta 2 microglobulin is for expressions. Now, in certain cases like your Dondi cells, okay, this is a kind of a tumor in which your beta 2 microglobulin is not formed at all. Okay, but your alpha 1, alpha 2, alpha 3, this alpha domain will be formed because they are being formed by different chromosome. Alpha किससे form होता है? 6 से and beta 2 microglobulin 15 से. So इसमें alpha तो form हो रहा है पर beta form नहीं हो रहा है. That means ये MHC1 molecule express ही नहीं कर पाएगा अपने आप को. Why? Because there is no help for beta 2 microglobulin. Okay, and यहाँ पे MHC1 molecule में which was binding to the CD8 of cytotoxic T cell, alpha 3, right? Because that is the only one left to function. And for this MHC2 molecule, it was beta 2, which was binding to CD4 of T helper cell. Now, don't get confused that beta 2 यहाँ भी है, beta 2 यहाँ भी है. This beta 2 is formed from chromosome 15, while this is formed by chromosome 6. These are two different. So, this is just for moral support. This beta 2 microglobulin here is just for moral support. Whereas in MHC2 molecule, from the chromosome 6, this part, the HLA gene complex, this part 2, the HLA gene complex, region 2, they form the whole alpha and beta chain. Okay, so this beta 2 is different from this beta 2. Don't get confused. Clear? Now let's quickly see the difference. Gene MHC class 1 has HLA-A, HLA-B, HLA-C. MHC class 2 has HLA-D, P, DQ and DR. Structures, it has one alpha chain, three domains, alpha 1, 2 and 3 plus beta 2 microglobulin. MHC2 has one alpha chain, alpha 1, alpha 2 and one beta chain, beta 1 and beta 2. Where it is expressed, MHC class 1 is expressed in all nucleated cells plus platelets. It is not expressed in RBCs and sperms. Whereas MHC class 2 is expressed only in antigen presenting cells like your macrophages, monocytes, B cells and dendritic cells. Must be doctor. It presents antigen to MHC class 1 presents antigen to CD8 cytotoxic T cell. MHC class 2 presents to CD4 helper T cell. Types of antigen, MHC class 1 only possesses endogenous from inside cell like viruses, tumor, whereas your MHC class 2 types of antigen is exogenous from outside also like bacteria and extracellular pathogen. Peptide binding group, where it was for MHC class 1, MHC class 1 is taking bite from a bread. So between alpha 1 and alpha 2 domain, MHC class 2, two friend holding hands. So the peptide binding group between alpha 1 and beta 1. Okay. Interaction with T cell, which part of MHC class 1 is interacting with T cell? Only alpha 3. Okay, alpha 3 is the one which was only functional left, and MHC class 2, alpha 1, beta 1 are busy with the antigen binding left. Alpha 2 is like, I won't do anything. So we have beta 2, which binds to the CD4 molecule of T helper cell. I hope it was clear. And now just quickly go through the revision. What all we have understood in this topic. MHC are genes coding for cell surface protein that presents antigen fragments to T cell, acting like display board of every cell. It is unique like Aadhaar card, okay? And since it's deciding compatibility between a donor and a recipient, it is called histocompatibility antigen. Since in humans it was discovered on leukocyte, it's called human leukocyte antigen, okay? Now, MHC genes are located on the short arm of chromosome 6 inside the HLA complex. MHC1 zone has genes HLA-A, HLA-B, HLA-C, which forms the protein HLA-A, B and C, which forms the alpha chain. And the beta-2 microglobulin is formed from chromosome 15, okay? And it is present on all nucleated cell and platelets except which one? RBC and sperm cells. Clear? And this MHC1, which is on all nucleated cell and platelet, it presents the antigen to CD8 cytotoxic T cell. Now your MHC2 zones had genes like DP, DQ and DR, which forms protein like HLA, DP, DQ and DR, which forms the alpha and beta chain of MHC2 molecule, which is present on antigen presenting cells. Which are the antigen presenting cells? Monocyte and macrophages, B cells and dendritic cell. Must be doctor. It presents antigen to CD4 T helper cells. And then we have your MHC3 zone, the HLA3 zone, which is like a support staff. Okay, what does it form? These proteins are complement, cytokines and stress proteins. Clear? Remember it like this. Your MHC1 is related to an assassin. MHC2 is related to advisor, and your MHC3, that is HLA3. There's nothing known as MHC3. It is the HLA3 gene complex, which is associated with accessory proteins. So 3A, assassin, advisor and accessory proteins. Thank you.